So, CRISPR, you've likely heard that name quite a bit, probably in connection with gene editing and all the ethical questions that come with it.
Absolutely. It's been a huge topic.
But today, we're actually diving into something else. Still revolutionary, but maybe less controversial. We're looking at using CRISPR to fight, well, superbugs.
Yeah, harmful bacteria, especially the ones that are resistant to antibiotics.
Exactly, using this biological tool to sort of turn the tables on them.
And it's really critical work. I mean, this isn't just some, interesting science experiment in a lab.
Right.
We're facing a potential crisis with antibiotic resistance.
Mhm.
If our drugs stop working,
It's a huge step backward.
a massive step back.
Mhm.
Think pre-antibiotic era where infections were, well, devastating.
Yeah.
So finding new ways is urgent.
And this CRISPR approach seems particularly clever.
It really is. It kind of flips the whole script. Instead of just like chemical warfare with antibiotics,
Mhm.
we're using the bacteria's own natural defenses against them.
Okay, so let's break that down. This idea of a CRISPR pill, how does that even work? How do you turn its defenses back on itself?
Well, first, maybe we should quickly recap what CRISPR actually is in nature.
Good idea. Because it's more than just a gene editing tool scientists invented.
Oh, definitely. It's a natural system found in bacteria. The name, CRISPR, it stands for clustered, regularly interspaced, short palindromic repeats.
Quite the mouthful.
It is. But basically, think of these little repeating segments of DNA in the bacteria. Palindromic means they sort of read the same forwards and backwards, like level or madam.
Okay, like genetic palindrome.
Exactly. And they're spaced out, regularly interspaced, with these unique bits of DNA tucked in between the repeats.
And those unique bits are key, aren't they?
They are the key. Because CRISPR, fundamentally, is a bacterium's immune system. It's defense against viruses.
Right. I think people forget that bacteria get sick too. They have viruses that attack them.
They absolutely do. It's a constant battle down there at the microbial level. So imagine a virus injects its DNA into a bacterium.
Trying to take over the cell.
Yep. But if the bacterium has seen that type of virus before, it will have actually stored a little snippet of that virus's DNA.
Where?
Right there, in those spaces between the repeating CRISPR sequences. It's like keeping a genetic mugshot of past invaders.
Wow. Okay, so it remembers its enemies.
It does.
Doesn't it?
And it uses that stored viral DNA snippet.
Yeah.
It makes a copy, but as RNA. RNA is like a little messenger molecule. So it makes an RNA copy of the viral mugshot.
Like sending out a wanted poster based on the stored picture.
Precisely. And this RNA molecule then floats around inside the bacterium looking for a match.
Looking for the actual invading viral DNA.
Exactly. And when it finds it, when that RNA wanted poster locks onto the matching viral DNA sequence,
What happens? The attack.
then the enforcer steps in. A protein that works with CRISPR, usually one called Cas9 CRISPR associated protein 9.
Cas9. Okay. I've heard of that one. The molecular scissors?
That's the one. Guided by the RNA, Cas9 finds the viral DNA and just cuts the viral DNA, disabling it.
Destroying the virus's instructions before it can cause harm.
Yep. Renders it harmless. It's a really, really ingenious primitive immune system. Simple, but incredibly effective.
Okay, so that's the natural defense. Bacteria using CRISPR-Cas9 to fight off viruses. But the pill idea, that's turning it inwards, right?
Exactly. So scientists looked at the system and thought, okay, bacteria use this to cut viral DNA. What if we could trick it into cutting the bacterium's own DNA?
Make it self-destruct.
Essentially, yes. Convince it to commit genetic suicide.
That sounds complicated. How do you deliver that instruction?
Well, this is where it gets really clever, almost like something out of, Game of Thrones or something. They plan to use a virus.
Wait, use a virus to deliver the CRISPR instructions to the bacteria?
Yeah, but a specially engineered virus. One that carries the CRISPR RNA blueprint telling the bacterium's own Cas9 system to target a vital gene in the bacterium itself.
Okay, but wouldn't the stomach just destroy this virus if you took it in a pill? Stomach acid is pretty harsh.
It is. That's a major hurdle. So the researchers came up with a kind of Trojan horse strategy.
A Trojan horse?
Yeah, they packaged the engineered virus inside another harmless bacterium, a type that can survive the stomach passage.
So you swallow the pill containing these harmless bacteria, which are secretly carrying the virus.
Right. They pass through the stomach unharmed. Then, once they're in the gut where the target harmful bacteria live, the harmless bacteria release the engineered viruses.
And those viruses then infect the target bad bacteria.
And deliver the CRISPR instructions that basically tell the bad bacteria, use your Cas9 to cut your own essential DNA.
Triggering self-destruction. Wow. That is quite a strategy.
It's pretty ingenious, using layers of natural biology against itself.
And one of the big pluses here, I'd imagine, is targeting.
Huge plus. That's maybe the most exciting part. Unlike our current broad-spectrum antibiotics.
Which just wipe out everything, good bacteria, bad bacteria.
Exactly. They carpet bomb the microbiome, which causes side effects and crucially helps drive resistance because all bacteria get exposed.
But this CRISPR approach could be much more specific.
Highly specific, because the RNA guide sequence can be designed to match DNA found only in the target bacterium. So it could potentially kill, say, just the harmful C. diff, while leaving all the beneficial gut bacteria untouched.
That's a game changer. Surgical strike versus carpet bombing, especially for fighting superbugs where resistance is the main issue.
It really could be. Preserving the good bacteria while eliminating the bad is, well, the holy grail in many ways.
But okay, let's temper the excitement a little. Where is this research actually at? Is this pill something we'll see soon?
Well, no, not soon. It's still very early days. This specific delivery method, the Trojan horse idea,
Yeah.
it hasn't even been tested in animals yet. It's proof of concept in the lab.
Okay, so still a long way from human trials or pharmacies.
A very long way.
Yeah.
And right now, they're focusing these early efforts on targeting just one type of bacterium.
Which one?
Clostridioides difficile, or C. diff, you might have heard of it, it's a nasty one, causes severe diarrhea, and it's often resistant to antibiotics. The CDC calls it an urgent threat.
Right, a major hospital-acquired infection too.
Exactly. But the challenge is, that virus they engineered to deliver the CRISPR payload, it probably only works against C. diff.
What is that?
Viruses tend to be very picky eaters, so to speak. They usually only infect one specific species, or maybe a few closely related ones. They're kind of species-specific assassins.
So you couldn't use the same virus delivery system to target, say, MRSA or some other superbug?
Probably not. You'd likely need to find or engineer a different virus that specifically infects MRSA, and then load that virus with the right CRISPR instructions. It adds layers of complexity.
I see. So scaling this up to fight many different superbugs isn't straightforward.
No, it requires developing specific tools for each specific threat. It's not a one-size-fits-all solution, not yet anyway.
Still, even with those hurdles, the basic idea is incredibly powerful, given how badly we need new weapons against antibiotic resistance.
We absolutely do.
Using the bacteria's own systems, its own evolutionary tools, against it, there's something almost, I don't know, elegant about it, turning its own defenses into the weapon of its destruction.
It is elegant. And it shows incredible ingenuity, finding these solutions within nature itself. The potential is definitely there, even if the path forward has challenges.
Absolutely. It sparks some real optimism in a fight that can sometimes feel quite daunting.
It does. And maybe, as we wrap up, it's something for you and the listener to think about. Facing this huge challenge of superbugs, what does it tell you about our ability, our sort of drive to science our way through these problems by understanding them, well, repurposing nature's own toolkit? It's quite something, isn't it?